Hello, my name is Juergen Heitmann. I'm the Chief Business Officer of Sensorion, a specialty company dedicated to the field of inner ear biology. And I'm going to present to you our company, our strategy, and our development pipeline. And being a publicly listed company, I have to share first this disclaimer. I'm going to make certain forward-looking statements, so please pay attention to this standard disclaimer. Sensorion is a company that is currently focused on building up a franchise of new gene therapy programs. We are doing this in close collaboration with Institut Pasteur. Institut Pasteur, led by Professor Christine Petit, based in Paris in France, is probably one of the leading academic institutions in the field of genetics of hearing. So we have initiated this collaboration almost 2 years ago, and since then have built up also our in-house capabilities. So we are currently run by an experienced management team in the field of gene therapy. Our strategy is to restore, to treat, and to prevent in the field of hearing loss. So apart from this gene therapy program, we also pursue a more standard small molecule approach. Based on these high-profile collaborations, we have also attracted high-profile investors. So apart from Institut Pasteur, we have a collaboration running with Cochlear, the market leader in cochlear implants. We collaborate with the French Army and also with Necker Hospital, the leading European pediatric hospital, also based in Paris. So we were able to raise last year significant funds, almost 70 million euros, led by investors like Invus and Sofinova. Sensorion was established more than 10 years ago. We went IPO in 2015, so we are currently listed on the French Euronext stock exchange in Paris. As we are dealing with a very specific field, I'd like to start with a very brief introduction or highlight on the inner ear biology. When we talk about inner ear, we talk about this blue structure in the center of this chart. It's located behind the earlobe in the skull, and it's a very delicate localized compartment. When you look into it, it's filled with liquid. It's divided in separate compartments, divided by membranes, and it comprises two senses. The snail-like structure is what we call the cochlea. It is hosting the sense of hearing, but it is combined with the vestibular system, which is comprising the sense of balance. So when you look into the center of this cochlea, you find in the so-called organ of Corti, highly specialized sensory cells. We call them hair cells. And they are actually the site where an acoustic wave, a physical wave from acoustic stimulation is translated into an electrical sensation by the sensory hair cells. We call them hair cells because when you look at them, on top of them, there is a bundle of tiny little hairs, stereocilia, that are detecting the acoustic stimulation, the acoustic vibration. One specificity about these cells in humans is that when we are born, we are equipped with a full set of sensory hair cells. It's a fairly small number of cells, maybe only 3,500 cells. They are fully up and running at birth, and they are meant to last for the rest of our life. So if we take good care of our hearing, we can maintain acoustic hearing until the end of our life. If we create any damage to these cells and they are damaged or they are actually dying, it translates into permanent hearing loss because these cells have no ability to heal, to regenerate. This is bad news for the field of hearing loss, but it actually presents an opportunity for the development of gene therapies, because if you suffer from a genetic defect and you find a way to correct this defect in early childhood, it will lead to a lifelong therapeutic effect. So this treatment will have a long-lasting or even everlasting effect. Our strategy is, as I said, to restore, to treat, and to prevent hearing loss. The restoration is focused on these gene therapy approaches. So here we are developing, in collaboration with Pasteur, various AAV vector-based gene therapies, namely against otophaline deficiency. It's the so-called DFNB9 deafness. We have recently announced new programs targeting GJB2-related hearing loss. So these are connexine related conditions. And we are also pursuing a program against Usher syndrome type 1. When we talk about treatment and prevention, this refers to our small molecule approach. So here we have a compound that has broad otoprotective properties and that enables us to treat acute hearing loss, a condition where these hair cells are damaged but are not yet dead, are not completely gone. So we can stop the apoptosis that has been induced. And if you know in advance that something may happen, an insult, you can think of preventing hearing loss. And here we are pursuing a medical condition called cisplatin-induced ototoxicity, where patients that undergo chemotherapy have a high chance to develop hearing loss, and giving our compound can hopefully protect the ear in this condition. The next slide is summarizing our development pipeline. So you see most advanced on top is the acute hearing loss condition. Here we are in the midst of a phase 2 clinical program. Full data are expected by end of this year. The preventive treatment, as I said, 3 applications, most advanced, this is cisplatin-induced ototoxicity. We are initiating a clinical trial in this indication by mid of this year as well. And at the bottom of the chart, the various gene therapy programs. You see most advanced is the autophagy deficiency program. We have almost completed the proof of concept in mouse models. And a little bit behind is the Usher gene therapy program against Usher type 1. And more recently announced 3 independent programs targeting GJB2 related conditions. And these 3 programs are targeting different patient populations. So we will initially focus on the adult segment, patients that develop an early onset presbycusis, so age-related hearing loss at an early stage in life. But there are also forms of progressive hearing loss in childhood and even congenital forms of kids being born deaf. Let me dig a bit deeper into the various gene therapy programs. So the autophagy deficiency is a condition where, due to a single mutation on the gene encoding for autophagy, is causing complete deafness at birth. This condition is probably affecting around 8% of all cases of congenital hearing loss. So that leads to a prevalence of maybe 20,000 cases in the US and also in Europe. GJB2 related hearing loss is much more common. So here we believe that mutations on this gene probably are responsible for 50% of all genetic forms of autosomal recessive non-syndromic hearing loss. Our focus will be on the adult population, where we see maybe around 100,000 patients developing early onset severe age-related hearing loss. So that means age-related hearing loss starting at the age of maybe 30 or 50 up to 69 years. The prevalence in the younger childhood population is even higher. We can identify around 200,000 pediatric patients in these conditions. Usher syndrome is a more rare condition, but unfortunately it's also the most severe form of congenital hearing loss. So these kids are born completely deaf. Their vestibular organ, which also comprises these hair cells, is also affected. So these kids have problems to learn to walk because their sense of balance is affected. And when they grow up at the age as a teenager, they usually also develop blindness. So a very severe condition, but more rare. There's very thin literature available on this, on the prevalence and incidence of these conditions. Maybe 4 to 17 people per 100 individuals suffer from Usher syndrome. Type 1 probably represents 40% of these cases, and we are targeting the USH1 gene in this condition. All these genetic forms of hearing loss, they can be identified early on in childhood. So in the developed world, we have a standard hearing screening for newborns. If these kids are diagnosed and confirmed with a genetic form of hearing loss, the only treatment currently available is a cochlear implant. So this is an electrical stimulation device that is implanted into the cochlea and is providing an electrical stimulation of the acoustic nerve. So it is not restoring acoustic hearing. It provides an electrical sensation that is currently the standard of care for these conditions. That's why we believe that gene therapy has the potential to provide really life-changing therapies in these conditions. I'd like to focus a little bit on the autophaline gene therapy program. Autophaline is an essential protein that you find only in the synaptic connections between inner hair cells and the subsequent neuronal cells. So autophaline is necessary to prime vesicles that are containing the transmitter in order to release this into the synaptic gap upon acoustic stimulation. So this autophaline only plays a biological role and is only available in inner hair cells of the cochlea. And if you have a defect in the sequence, leads to a non-functional autophaline protein, it completely disconnects the system. So the vesicles are no longer able to release the transmitter and the whole system is deaf. But there is no other condition in these hair cells that is affected. So that means a simple restoration of an intact protein has the potential to reconnect these cells and to restore acoustic hearing. So that's why we believe this represents almost a sweet spot for gene therapy, because you only have to deliver the corrected sequence to a very small amount of cells in a local isolated compartment. And as I mentioned in the beginning, if you successfully convert these cells, it will lead to an everlasting or potentially everlasting therapeutic effect. Institut Pasteur has identified this condition and has generated a knockout mouse model. What you see here in green glooming is the autophaline present in inner hair cells. On the left side, you see the normal condition in a normal mice. The knockout mouse is not expressing any autophaline, so the cells are completely dark. And after a single injection, the protein is again expressed. And they could also show in the corresponding electrophysiology that the cells are electrically excitable again. The challenge for these gene therapies on autophaline is the size of the sequence. So it does not fit into a standard AAV capsid. We are splitting the sequence in two compartments, delivering two vectors, and we have shown that that works. So the sequence is successfully reconstituted, and it leads to the expression of a full-length autophaline protein in the target cells. The status of the program is, as I said, the proof of concept in the mouse model has successfully been completed. We have already announced preliminary non-human primate data, and we have secured a CDMO partner with Novacep. Currently, we are discussing our proposed development plan with regulatory authorities. I'd just briefly like to mention the other two gene therapy programs. Connexins are also proteins that are essential and endemic for the cochlear tissue. We are talking connexin 26 that you only find in the cochlear cells, where it creates the gap junctions, the connection between neighboring cells. And for Usher, Pasteur has also generated a nice proof of concept. This is published data as well, where they were able to restore the anatomy of the hair cells and also the function in knockout mouse models. With this, I would like to finish, and I'm happy to answer any questions that you may post in the chat room. Thank you very much.